Recording. Oh, okay. Yeah, I think we can start now. Hi, everyone. Welcome to this week Machine Learning for Protein Engineer seminar. And this week we have Jana from Stanford University. She's going to talk about her recent project, zero-shot design of a de novo metal enzyme. And you can take it from here.
Yeah, thanks, Bo. I'm really excited to talk to you guys about this really long project, actually. It's my main thesis work on the zero-shot design of a de novo metalloenzyme. So I guess to preface a little bit why we will care about enzymes in general and just how difficult they are. So enzymes catalyze these really difficult reactions. And so their uncatalyzed rate, how much time it takes for them to catalyze these reactions varies anywhere from 5 seconds to 1.1 billion years. But all of the reactions we have here listed are all reactions that enzymes naturally do within our body. And so many of these are facilitated by, for example, like the pancreas or engineered enzymes like petases or even proteases. And so um, something like proteases takes about 450 years, and that's nearly half a thousand years. Or you would wait for like a Mendelic racemization reaction, and that would be from like when the Giza pyramids were last built. And so these are really, really difficult reactions. But what enzymes are phenomenal for doing, so something like peptide hydrolysis, is that you have this enzyme, and sometimes it has this metal center, and it brings that time down, that half-life down to about 30 minutes. And so enzymes do this across the board on all of these reactions, making them far more efficient and also very specific for things in our daily lives to continue to be carried out. And in other words, what that really means is that it's lowering the energy barrier. And so you go from a reaction to a product and you have this really massive energy barrier, and then you get an enzyme, and that enzyme actually lowers that energy barrier, making it a more accessible reaction. Um, Cool. And so, like, one of the fundamental questions we like to ask about biology broadly or designing protein engineering tasks is what if we could build biology to do the exact chemistry we want it to do? And so there's many chemistries where we don't have enzymes that we want to be able to catalyze these very difficult reactions very specifically. And so how do we actually do that? How do we do it with really high efficiency? And how do we continually have an enzyme able to turn it over? And so that leads to two really fundamental questions. The first one being, how do natural enzymes perform their functions? And then what do we actually need to learn to design them for new functions? And so this is actually more of a fundamental question of like, where do enzymes get their catalytic power from? Um, so I'll walk you through the very first enzyme, metalloenzyme that was discovered, which is carbonic anhydrase. And it is also the very first enzyme that I really like sat down and tried to understand and study. And so it was discovered in 1933, about 100 years ago. And it has this zinc metal center, and it catalyzes the carbon dioxide and water to bicarbonate and proton. And this is really important for regulating pH in our bodies, and then just in animals in general. And so this does the CO2 hydration, which like uncatalyzed without an enzyme takes 5 seconds as a half-life, but carbonic anhydrase makes this 10 to the negative 6 of a second, so really, really fast. Um, and so in a lot of de novo design tasks, what we would consider the catalytic motif, or what is typically defined as this catalytic motif, would be something like the three histidines that's coordinating that zinc. And so this would be considered like the direct residues involved in the catalysis for this reaction for CO2 hydration. But one could also consider the active site residues, which would be the carboxylates that are mediating the water, allowing actually for this whole reaction to take place. And so these are really started to be understood about 50 years later from when they were discovered in 1988. But if you in 2007, and I think in a lot of like previous and after that literature, there's are these second shell and third shell residues, and they really make up this like really tight network of hydrogen bonds, both interacting with water and with other residues, all coming back to where that catalytic motif or that zinc center is. And so these are involved in things like conformational gating and proton shuffling and just the like rigorous hydrogen bonding network. And so if you start mutating any of these residues that are colored in orange, you reduce activity by over 1,000-fold, or you would kill the activity entirely. And so the catalytic motif or those active site residues are not the only things that are really important for catalyzing these reactions. Um, and so achieving catalytic competence is much more than just the motif for those active sites. And so we actually need those second shell and third shell residues. Things like the catalytic motif and active site residues are actually now how people typically do this design problem, where you would take this motif from a natural given scaffold, and then you would put it in to a new de novo scaffold with something like RF diffusion, and then you would design around it and screen hundreds. And what that really misses is all those second and third shell residues that are really fundamentally important for that catalytic competence. And so Ariel Warshel and Michael Levitt and Martin Karplus won the Nobel Prize in 2013. And this was really on that fundamental fact that enzymatic activity depends on both the chemistry, the geometry, and the electrostatics in the active site. And it's not just the amino acid identities or their orientations that allows for catalysis. And so that is actually just a fundamental thing that we want to keep in mind when we try to design enzymes, especially when we want them to be as reactive as things we see in nature. And so John Jumper and David Baker in 2024, so 11 years later, won the Nobel Prize for protein structure prediction and protein design. And we now can really just design like thousands of de novo proteins at the click of a button. And really the question that we want to ask is how do we make those functional, not just small sporadic functions or directed evolution? How do we actually make them out of the box at the click of a button, functional like the ones we see in nature? So we can take a lot of inspiration from how nature evolved enzymes. And so enzymes really evolved to be highly specific in the environments that they occupy. And so different species will evolve different amino acid sequences to do the same type of reaction in that same enzyme scaffold, that native enzyme scaffold. And that's where we see a lot of sequence diversity. But the thing we typically see that is conserved is the active site, the amino acid identities, and roughly the chemistry in that active site to be actually able to facilitate those reactions. And similarly, we see something with cellular localization. So depending on where an enzyme is in the cell, it'll adapt to be able to survive that cellular space, but the reaction still can proceed, and that's because the chemistry itself is conserved. And so similarly, like enzymes have also evolved for different functions. And so you'll see enzymes that are very related through this like ancestral reconstruction plot. But somewhere in the back in that reconstruction plot is what people consider an ancestral enzyme, which is this enzyme that was able to originally catalyze both of the reactions and then over time in evolution has made it highly specific and highly efficient for a very specific reaction. And so these are some things to keep in mind of like how nature came about solving this problem, and it did it with years and years of selective pressure. And so now the better question is, can we evolve for function in silico where we really want to get at the chemistry that we're looking to tackle? So we can take inspiration from evolution. And so evolution did this by natural selection or that selective pressure. And so what will happen in evolution as we study in like biology in high school is you'll get a population, and this population will then just be evaluated. And so some will survive and some will not. And then this selection will then occur, and so then they will reproduce and something will mutate, and then they'll make a new generation of population, and this will continue on and how evolution, we can think about it in the human sense, occurs. And so we, with Simon, who was visiting our lab a couple of years ago, we kind of came up with this plot where we do this analogous genetic algorithm type of thing to actually be able to evolve for a protein scaffold in silico. And so we start with a given structure, and we can initialize that structure with like 10 different sequences, 20 different sequences, 50 different sequences, and we evaluate it basically along the criteria that is most important to us. And that can be anything from can it bind based on my predictive model to how many hydrogen bonds does it have. And so these are just like functions that we can come up with. And so we'll then select for the elite, and this will be following the next, the generation algorithm, the genetic algorithm. And then those sequences will cross over similarly to how we see reproduction happen. And then we'll generate some random mutations, and then we'll reevaluate the population. And we can do this iterative cycle, hoping to actually evolve in silico for things that are optimizing multi-objective scoring functions. And so what that actually looks like in principle is you have the starting structure and it might have a really good sequence for that given backbone, but really fail at that second objective that you care about. And then over time, it'll do this mutation and crossover. And then eventually we'll reach an optimized structure where the structure still has the sequence that fits onto that backbone, but also is highly predictive to be for the function, the objective that you're looking for, that second objective. And so we really want to like an optimal maxima of the both probabilities, or in any case, you can go up to 20 probabilities for how we've kind of come up with this. And so we really, in terms of designing or evolving proteins in silico for something like an enzyme, what we're really looking for is a sequence, a structure, and a chemistry that all fit that given protein. And that's a much more difficult task. And so we really want to eventually in some world, we want to optimize probability of sequence and structure and chemistry. But that's a little bit more difficult. And so we can instead kind of break it into two probabilities that are a bit easier for us to model in terms of machine learning probabilities. And so we have these two probabilities. One is the probability of the chemistry given the sequence and the structure, and the other one being the probability of the sequence given the chemistry and the structure we're looking for. And so we can then address these probabilities with a deep learning model. One, a deep learning model that predicts the chemistry given a sequence and structure, and one that predicts the sequence given the chemistry and structure we're looking for. And so eventually over time, as we sample those two probabilities, we'll find this like jointly optimal solution between the two. And so we call this dEVA, so designed by evolutionary algorithm. And so the way we have it kind of built out is that you'll define the objective functions that you're interested in. And so this is as simple as basically returning some numerical value for whatever property you're interested in. And then you'll identify some candidate backbones. And because it's so easy now to make structures at the click of a button, you can generate thousands, you can subset, you can say these are my target scaffolds, this is a target region. And so just identify those backbones. And then you'll do this joint optimization with this genetic algorithm. And then eventually you will then either find an optimal design for that given scaffold, or it will fail in that search space. And so you can try again with a different backbone. And so we have this question, and then this chemistry is really, really broad. And so we decided to actually narrow it down a bit to metals. And so the first question being, can we design metal binding proteins? And then can we design metalloenzymes where the metal center is responsible for the catalysis? Metals in general are really important for other things besides catalysis. And so they're actually very important for stability and also ligand binding. And so they're very ubiquitous in nature. And so the way that we decide to address this is that we have this probability of metal given the sequence and the structure. And so this really is responsible for metal site design given some amino acid environment context. And so we use metal 3D, which voxelizes in a 3D CNN a 20 angstrom cube. And we'll then run this through the CNN and predict where a metal would be and with what probability given that voxel environment. And we can do that across an entire protein. And then to do the probability of sequence given the metal and the structure, we're then really looking for sequence design given some ligand context. And so that would be something like ligand MPNN, where it was trained to then predict a sequence if there are other ligands, including metals, in that atomic environment. And then additionally, also with the added advantage that it builds in rotamers or models and potential rotamers. And so the biggest assumption you make when using this design by evolutionary algorithm pipeline is that you're assuming the models you're using are good approximators of the respective probability distributions that you're after. And again, models can be anything from deep learning model to just like a mathematical function. And so over time, you'll start at like some given scaffold. You'll see start with a given scaffold, and it will initialize something that is actually fairly poor. And so something here like I, where you have two carboxylates mediating one zinc, is actually predicted pretty poorly. And then over time, as you're doing this sampling with the genetic algorithm, you're slowly starting to explore more plausible metal binding environments. And eventually, it actually finds at this optimal Pareto front this 2-cysteine, 2-histidine solution. And that would then be your optimal design to then purify and test. Oops. And so this is actually the plot for that exact protein, this immunoglobulin. And so you actually do have this 2 cysteines and 2 histidines, and you can see that it's like at the Pareto front, at the top right corner, so that apex. And so here you have a comparison to a zinc finger motif, the closest zinc finger motif we can find in the PDB, and the motif RMSD is 1.61 angstroms, and that's fairly far. The rotamer environment is actually quite different than a typical zinc finger. And so we can consider this actually a de novo motif where we were able to design this binding site for a metal, but it actually looks nothing like in terms of like the rotamer orientation or the side chain orientation to anything we see in the PDB. So this would be actually the first instance of something like this in the de novo design space where we're not stealing a motif from nature. And so I actually purified all of these proteins, and we can do a competition titration with Magfura. We see it actually binds metal quite tightly with a kAD of 37 nanomolar. And this is on par with many natural metalloproteins. If you look at what it does to the protein, we can try and we can run the CD and melt the protein in APO without any metal, and we see that it actually is fairly unstable, thermally unstable without any zinc. But when we add zinc, we see that it retains some stability a bit more. So the zinc here is modulating thermal stability. And then with Koei and Reedy, we can run XAS, which really defines the coordination environment of a metal inside the protein structurally. And we see that it's actually the spectra, the EXAFS data, is quite distinct from reference zinc fingers, where this experiment's been done before. And so this gives us some indication that actually the motif in solution is really distinct from natural motifs from what we see. And so this is really exciting. And we did this design for 8 different distinct metalloproteins of varying sizes from everywhere from 90 amino acids to 250 amino acids. And so here's another example. Well, with Matthews, who solved the crystal structure, where we see in this asymmetric beta propeller, this is a novo beta propeller, a high resolution crystal structure where the metal is in the coordination environment that we're expecting. And so this is all really exciting. Now we're able to design this out of the box with only one protein sequence per structure. I didn't get a chance to say this, but none of these designs actually ran through AlphaFold or any structure prediction model before we ordered them. So this is purely based off of where it is along the Pareto front. So here's another example. So whenever I like to do design problems, I always have a built-in negative control where I'm really testing at the hypothesis that I'm looking for. So this is this negative control where after the fact, we predicted with AlphaFold 3. These designs were all ordered before the release of AlphaFold 3. And so we see the structure matches with the design that we have. But we get a really high PLDT for the zinc at where it's being coordinated by just one carboxylate. And if you know anything about metalloproteins, zinc is typically coordinated with 4 or 3 amino acids. And so this is actually a non-productive binding site. So Metal3D predicts this with a probability of 0.56, whereas the zinc PLDT from AlphaFold is 87. And so that's really high. So for all distinctions, dEVA kind of predicts that it won't bind, and AlphaFold thinks it will bind based on the probabilities. And so indeed, when we do the competition titration, we see that there's no binding affinity. And then I also additionally check this with mass spec with the reference metalloproteins that we design, and we don't see the metal anywhere in the mass spec. So there's no zinc in the mass spec, and so there's really no detectable binding. There's no zinc coordination in this space. And so really this lends us the question of like, why is AlphaFold 3 predicting zinc there with one carboxylate? And so ML models learn from the data they're trained on. And so the good question to ask is what was the data it was trained on? So we took all the PDBs. I took all the PDBs that it was Metal 3D and Linked and PNN were both trained on, not wanting to do this across all of the PDB, but a subset where the models that we were designing with were seeing these metal sites. And the first thing we noticed is that 10% of the residues have no metal within 3 angstrom, or sorry, no amino acid within 3 angstroms of the metal. And so these are just free floating metals in the crystal that is in the PDB. Um, and so that's concerning. The next thing we see is that over 50% of the metals in the PDB have 2 or less amino acids within 3 angstroms. And so 1 and 2 amino acid coordination environments like I said, a non-productive metal environment. So this is also additionally concerning. Um, and then the thing we can see is that actually about 12.5 to 15% of the metals in the PDB have just one carboxylate coordinating the zinc. And so that's actually very reminiscent of the original negative control that we were looking at with AlphaFold 3, where we just have one carboxylate within 3 angstroms. So why are the data sets like this? If we know that biologically these are non-productive. And so actually the machine learning models are all learning off of these crystallographic artifacts, and that kind of sucks. So here are 2 examples in both the ligand MPNN and metal 3D training data sets. 2EJC, which has 26 zinc atoms in the structure, many of which are overlapping zinc metals, many free floating with no atoms within a reasonable distance of it. And then below 3IVB with a lot less, but now we have actually 2 zincs basically overlapping in the densities. And so these are a result of the crystallographers actually crystallizing the proteins in solutions that contain 1 to 2 millimolar of zinc sulfate. That's actually just a crystal additive to help us resolve the underlying protein structure, but is not relevant to the actual biological protein. But all the machine learning models, including AlphaFold 3, trained on this, and so these are just built in as things that are realistic. And this includes like an MPNet and Metal 3D. So these are not exceptions. So if we actually try to identify them, basically remove all the metal sites where there is 2 or less amino acids within 3 angstroms of each zinc, we actually see this really nice trade-off. Where the recall for those 0, 1, and 2 counts decreases significantly, but retains the probabilities for both the 3 and 4 amino acid environments. And so machine learning models can learn from better data, and they're still doing just as well. So that doesn't really affect so much what we're looking at here. I couldn't run this for ligand and PNN, unfortunately, because the training code is not out. So anyway, so then now we wanted to ask the question of what if we took this probability of metal and changed it to catalytic metal, and so making a catalytic metal site. And so what we really need now is a model that predicts the location of a catalytic metal within that atomic environment. And so Mahomes, too, from Jan Slutsky's group, had this really beautifully curated training data set for all the metals, identifying whether they're enzymatic or not. And so we can filter them to only zinc, and we get 2,177 total proteins, and then about 285 of them were labeled as enzymatic. And so this total 2,000 was a full training data set. And if we look at the ones labeled enzymatic, what kind of reactions are they catalyzing? We see that they're across the board all 6 enzyme classes, the majority of which are actually these hydrolyses. So they make up 56% of it. If we look more deeply at what bonds are being cleaved in these hydrolyses, we see that about a third of them are for these phosphate oxygen bonds, so this phosphoryl transfer type of reaction. And then of those in the active site, there is either 1 zinc, 2 zincs, or 3 zincs. And so we can see there are bi and trinuclear sites within these training data sets. And so now we can train metal 3D cat, so catalytic metal 3D, on this new filtered Mahomes training data set. And so now we have a model that predicts probability of catalytic metal given sequence and structure, and we continue to stick with ligand and PNN for designing the local atomic environments. Cool. And so now if we go back to this wheel, what we're really looking at is the two objective functions here are metal 3D cat and ligand MPNN. And then these are used here as our two probability A and B. And so actually for the enzyme design, I decided to basically minimize our protein search to just Tim barrels. And so we used very overfit Perpirdelli, the original Perpirdelli, which basically had an affinity for Tim barrels and made a massive library and filtered them down. So we had 20 total backbones that were Tim barrels, and then ran this design in those inside the core of the Tim barrel with dEVA, and then chose the Pareto optimal designs from there. And so from those, I ended up with three design A, B, and C that actually reached that Pareto front, and those were the three that I purified and then tested. And so the one that's most notable is this design B, which was actually designed without me specifying one, two, or three zincs with two zinc ions. And here we can see that it actually catalyzes this phosphate monoester hydrolysis, much more above background than the other two designs, and then the blink. So really excited about this, and so decided to see how real this was. So the first thing is looking at the structure itself that got designed. We see that there's this really beautiful environment. So one of the zincs is coordinated by three histidines, and the other coordinated by a histidine and some carboxylates. And so if we search for this Tim barrel, there are no structural neighbors within full-seq, the closest one being a TM of 0.69 was actually a de novo Tim barrel, also from Posu's group. There are no active site neighbors via motif search or full-seq. So if we take those amino acids and search for them in the PDB, we don't find any with similar rotamers and also amino acid identities. And there are no sequence homologs. So the closest one is at 18% sequence identity. And so this really is like a de novo protein, despite being within a Tim barrels which don't actually have any zinc phosphomonoesters naturally that are close to this. We can actually just test whether it is actually indeed metal dependent. And so we confirm that there are two zincs with mass spec. And then if we add EDTA, we see that it significantly reduces activity over time. And then we see that actually is turning over. So it is a metal dependent catalysis. If we mutate each of the individual amino acids and then double mutants and then triple mutants, they are all important for a catalytic function. And so mutating them away into alanine actually does reduce the catalysis. So we're in the right active site environment. And then I don't have this now, but we also have a structure as of this morning that also confirms the amino acid identities for those two zinc ions. We can do these pH titrations, and so we can titrate with we can have different pHs for the enzyme and actually catalyze and actually check how they're catalyzing the reaction at what rates. And so we see that there's this optima at alkaline pHs, and this is really consistent with this bizinc mechanism, where this is the optimal pH for activating a hydroxide and then actually allowing for the substrate to kind of bind and then allow for the phosphoryl transfer. And so now we're a little bit more happy, I think, or understanding of the mechanism at play. The protein does turn over. It doesn't just die after one reaction. And so over time, here's at like substoichiometric zinc, we can see that it continues to turn over and doesn't plateau. We did this with the Koe and Reedy. We actually went back and checked in excess. So what is the active form of the protein? So phosphate here, PO4 3 minus, it acts as a transition state analog, actually, where it's then binding to the two zinc ions. And so we can actually get the zinc-zinc distance, and then also the zinc phosphate distances, and then the nitrogen distances from the zincs. And we see that they're actually fairly consistent with the design model that we have. And we can confirm that there is actually phosphate binding at 1-to-1 ratio with a malachite green assay. And then we can also check inhibition assays, and we see that the protein does get inhibited by zinc. It has a Ki apparent of about 1 micromolar, which is also consistent with many phosphomonoesters that exist in nature. And so I never got to the kcats, so this is really exciting. So the design enzyme is also really efficient. We have a kcat over km of 1300, but if you put that in context with the uncatalyzed reaction, that's a kcat over k uncat of 10 to the 8th, and then a kcat over km over kw, or otherwise a catalytic proficiency of 10 to the 13th. The rate enhancement is 10 to the 13th for this model substrate. And so all of these enzymes that we have here um, are natural enzymes that have been characterized with this form up. And so we can, or PNPP, which is the absorbance analog, and we see that it actually has a kcat over km that's comparable to many of these natural phosphomonoesterases. So this is the first of its kind in that regime. And we can actually look then at just like different substrates. Is it catalyzing them? And so diff-MUP now here has two fluorines in addition to the entire substrate. And so this changes the chemistry and also the pKa of the substrate. And we still see a very consistent kcat over km and a kcat over k uncat. And we're really excited that it's not super specific to form up. Again, like this protein was not designed to have specificity. And so the next question I wanted to ask really is, does it have in this phosphoryl transfer reaction group, can it catalyze other phosphoryl transferases? And so here we have phosphodiester hydrolysis. And so this is bis PNPP and methyl PNPP, and it has a kcat over km of 1.2, which is actually on par, if not a little better, than many natural phosphomonoesters on the same reaction, and which is also a rate enhancement of 10 to the 11th. This is a very, very difficult reaction. Phosphodiester hydrolysis is the reaction that many RNases and DNases actually catalyze, so breaking DNA, and we want that to be a very difficult reaction. And so to put this more into context with the plot that I showed at the very beginning, phosphomonoester hydrolysis takes over 500,000 years, and then phosphodiesterhydrolysis takes over 13 million years to catalyze spontaneously. And so now we have this design B from this study. And so we can see that phosphomonoester hydrolysis takes a half-life about an hour, and then phosphodiester about a month. And so those are significant rate enhancements. And so this is the first de novo enzyme designed without stealing a motif from nature, but trying to actually catalyze these reactions with a rate enhancement of greater than 10 to the 13th. I think most of the ones that have been published so far are within the 10 to the 4th to 10 to the 6th. For metallohydrolases, it catalyzes phosphoester hydrolysis. This is the first example of such a protein catalyzing such a difficult reaction, and it was made in 0-shot. So this design did not run through AlphaFold filtering. Had I run it through AlphaFold 3 and bolts, one would probably not have ordered it. And so this is the example of the coordination environment that we see from these predictions, which are both from AlphaFold 3 and from bolts 2. And we see that they're actually very inconsistent with what we see in both the design and then also from all the experimental data that we see structurally. And so this is maybe then begs a couple of questions. Do we really need structural prediction to be so heavily incorporated in our design protocols? Some like bigger, broader questions are, how do we really optimize for catalytic chemistry? How do we minimize for screening these designs? So in this instance, I only screened a total of like 11 designs. I found that like the side chain modeling here is actually incredibly important. And so a lot of the positive results here came from good modeling of side chains. And this is a really interesting like example of designing a very promiscuous catalyst, very similar to how nature or evolution have designed starting enzymes and then evolving them to be highly specific, but also as just like building blocks for many different reactions for different specificities. So is this actually a more interesting approach or another interesting analogous approach for further protein enzyme engineering? Um, so anyway, I had a lovely team across Stanford, Slack, and EPFL, and then all of the facilities at Stanford that have basically done various parts of the characterizations with me, especially like the mass spec and the crystallographies. So really thankful for all of those. The bioarchive's been out for about a month, and hopefully we'll update it with the crystal structure soon. And then the code is also available on an MIT license. So feel free to try it out. And then if you have trouble, let me know. Should be easy enough to add multiple objectives, the whole assumption being your objectives are good. So thank you so much for listening, guys. And if you have any questions, I'm very happy to take any.
Well, thank you for the great talk. If you have any questions, then feel free to just unmute yourself. Oh, I see a hand raised. Also, you can unmute.
Oh, I can look at the maybe I'll take people who want to unmute. Maybe I can also take the ones in here. What was the process for cleaning the metal PDB data set for retraining metal 3D? Super simple. Just find all the zinc atoms that are in the PDB structure. So just load those in, and then find any atoms within 3 angstroms of that metal and just identify those. And so if and then I calculate how many of those belong to how many different residues. And so if there's two or less amino acids within that zinc, I just ignore that zinc. So there's just like a second text file, and that one just gets ignored, that specific zinc atom. And so very simple, uh, very simple. It's just very naive and very simple. And I'm sure there's better ways of doing it based on like analyzing how that protein was crystallized, or if there is a good enough density there, or if there's anomalous data. But I did the really naive, simple thing. Um, does your code allow for chemists to edit the scoring functions? Yes, it's actually plug and play. And so if you go to the GitHub, you should be able just to make a Python file. You need an initialize function and you need a score function that returns a float value. And then you just run that one line in the run.py and it should just out of the box work. And if that doesn't work, let me know. But I have a couple example scripts, and so hopefully those should be easy to follow along. I think Claude can also follow along because the README is fairly verbose. So just feel free to message me if that doesn't work. Both models, ligand and PNN and metal 3D, require protein structure input. During the design phase, do we still need to provide a structure? If so, what structure should be used as the input? Yes. So this is a structure-based approach, not a sequence-based approach, being that we're trying to like optimize the local atomic chemistry. And so those are like the side chains, for example. And so yes, you do need protein structures. And so I use the fact that they're in tin barrels, the cavity is really well defined, and I know roughly that I want 2 zinc atoms or 1 zinc atom or 2 zinc atoms. So I want them to fit in some pocket cavity. And so I just like design thousands, I think with Propertelli, because it takes like 10 minutes to make 1,000 unconditional structures and then clustered and then chose a subset. So that's how I decided to do it. You can choose them based on a priori knowledge. You could even motif scaffold and use that as a starting point, if that's like a good candidate, and then optimize it in silico where you have a starting point there. So really, it is just like a completely up to you. I think scientist intuition is just as important here as just having out of the box. But I think it can also be user dependent and problem dependent. So happy to also help out there. So feel free to reach out. Why are AF and Boltz structural predictions very different? That's a great question. I feel like the Boltz team can probably address that. I don't know about the AlphaFold team. My guess though is that it's very hard to predict where metals are within these structures, because especially within the barrel that I had. Um, wait, let me go back. Within the barrel that I had, there's quite a few carboxylates and histidines, and so the side chain orientation here plays a very important role in how it gets coordinated. And so something like AlphaFold, ooh, I can wait, I'll show you. Something like AlphaFold, this would be something tetrahedral if that is how the amino acids are actually coordinated. Similarly here, like this would be considered tetrahedral, because you would have like a water here. Um, and so those could be somewhat valid. Same here. But I think it doesn't take into account all of the like the context within the protein. I think it's trying to place them. Also, AlphaFold 3 and Boltz 2 were both trained on unclean data. And so they are also like tuned to also be highly predictive of a metal binding center being next to just 1 or 2 carboxylates. So this might also have an effect here. But it's a really good question. I'm just really glad I didn't use either predictive model before as a filtering step. Why? Okay, sorry. Yeah. So those are just my observations. I don't know. And I think that's a really great question. Um, okay, those are the ones that are in the chat. If anyone wants to unmute, I promise I don't bite.
So I have a question on like if you could also optimize or make like a organism specific variant of these designs. So like if you could integrate in to the like your scoring function
Hmm.
here to like make sure it's like also optimized for like the E. coli environment or like a different organism's environment. And
Yeah.
I'm thinking along the lines of like could you evolve like a different like metabolic pathway in a different organism maybe.
Metabolic pathway and then optimize in a specific environment are two very one has a far
Yeah.
more impact just than the other, right?
Yeah,
It
one leads to the other is what I was thinking though.
Yeah. No, that's a great question. So I guess I glossed over some of the design choices that were made in my designs. And so I actually seeded the outer barrel, like so outside of the tin barrel where the helices are, with a Calabi sequence. And so I chose a Calabi sequence. Those are highly charged, really soluble. But one could, in principle, also design the scaffold with like a fine-tuned language model or something related.
Mhm.
Um, and that should, in principle, have a surface area that is tuned for the environment that it's in. And you can probably also just fine-tune said language model, like an ESM type of thing, on like a subclade that you're interested in. That would be one approach I can think of. Because in chemical environments, in principle, like the one that what's doing the chemistry should still retain itself unless you're incorporating dynamics, and this is a whole other um downhill
But
slope.
there's also like different pH ranges it might perform best at, like where like different cytosols have like different amounts of salts and like
Oh yeah.
osmolytes and such.
Yeah, I mean, I guess it's just really dependent on your predictive model. So you could imprint, like, I could in principle be like, oh, I'm gonna predict my metal center thingy only protein enzymes that are annotated in UniProt to catalyze reactions at pH 4 and lower, right?
Hmm.
And then that would learn like local metal environment for that specific pH range. Um, so that could be one way of doing it. You could also like I guess tune it for a specific like enzyme, like if you had like a specific enzyme type, um, like you only want to do phosphomonoesters and only learn phosphomonoesters. So that would be
Mm.
like the opposite avenue between species and like reaction.
Mm.
I don't know. These are just like ideas, right? So this is like the nice
Yeah,
thing is that this is like plug and play sort of thing. So it's like you build something and then these models are not big. So like you can train these models like on your laptop without having even like if you have a MacBook with a GPU on it, like that should be sufficient to train it. Because like most of the models that I presented here, besides Ligon and PNN, were trained in 4 hours. Like they're super tiny models. The model weights fit on GitHub. That's how tiny they are.
yeah.
Uh,
That's really cool.
that is my criteria for small model. So like the nice thing is that you can have really tuned like predictive models here. Um, yeah,
Great, thank you.
these are
And
my ideas.
thanks for the talk as well. Really well presented. Thank you.
Thanks, Austin. Any other questions?
Cool. Sorry, I have a question. Is it
Yeah.
reasonable to like template the model when you use both or AlphaFold 3?
For
Because
like like a starting structure?
yeah, because
Yeah.
look at the predictions, like all the scaffold are like vary a little bit compared to the design ones. Maybe
No,
it's
so the side chains vary a lot. So the active site here is actually what varies a lot. So
Oh,
not even looking at the tin barrel, but looking at the actual active site, these are not like, at least to my eyes, these don't look like productive catalytic environments. And usually for at least like bizinc hydrolases, you have one metal that is very tightly bound, and this is typically considered like a structural metal, and then you have one that's a bit more weakly bound. And so in my case, the one with the three histidines is tightly bound, and the one with the carboxylate and the histidine is more weakly bound. And this is actually what allows for that mechanism. If I run metal 3D on the metal sites for AlphaFold 3 and bolts that I'm presenting here, those don't actually have a good probability of being catalytic. Um, so the side chains, at least in this space, matter a lot because I have at least my models are tuned to be very sensitive to the side chains.
Yeah, got it.
What is the origin? Oh, that's a very deep question. What is the origin of kcat over km? Is it the proper placement of both zinc or is it substrate binding? What you say the high kcat over km is surprising or is it expected? Oh, that's a great question. Um, So let's see. So the Km here is actually quite tight. And so the Km of 0.29 is really tight. The Kcat is actually what is slow. And then the structure isn't out, and I don't have a slide with the structure, but I think it's because the Kcat is actually limiting in this sense because you need a catalytic water. And so the bummer is that you can't model in water. You need this like hydroxide that gets activated between the two zincs, and this stabilizes and actually allows for the transfer. So what you can imagine is that the phosphate binds very tightly, and we know that it binds tightly from just the inhibition titrations. But how long it takes for the water to actually be stabilized between the two zincs and then allow for the transfer is limiting here. So substrate binding is good. Kcat is like poor, and I'm like proud to admit it. The Kcat over Km is a trade-off between the two, and that's the relative efficiency. What is more, I think, uh, so I think that the high kcat over km is like reasonable to what is expected and what we get from these designs and what we can understand from the chemistry. Because there's no residue that's coordinating the water. That's probably just like a flaw of like how this design process where it was like trained on everything. Um, what really is like both surprising and also unsurprising is its proficiency, so divided by the rate of water. So we have 10 to the 13 here. And so this is a very difficult reaction. So uncatalyzed for just this like model substrate is 10 to the negative 12. For like some natural like phosphoesters that were people like that your body or many natural enzymes hydrolyze, that can go up to 10 to the negative 21. So this is a very difficult reaction. And so the rate enhancement as a result is both exciting and pleasantly surprising, and then maybe expected because we do have this bizinc mechanism, which is very stable. It holds the substrate, and like we're just basically waiting on the water. So something you could do in terms of like a directed evolution approach is can you optimize where that water is, that hydroxide, to do the full cleavage? That's a very long-winded answer. I hope it answers your question. Can you go over the evolution part again? You mentioned optimizing, you mentioned that you were optimizing for the two probabilities. Yeah. Um, cool. So maybe I go, where do I go? Maybe I go here. So what happens in the evolution model is you start with this like given structure, and so you can make this however, like you can use AlphaFold to predict a protein and then use that. You can use RF diffusion and make some. You can use Properdelli and make thousands and run this. And so you have a starting scaffold. You could have a Tim barrel, and so you initialize the sequence, and so you can initialize the sequence with whatever like model you want. So you can run protein MPNN on this. You can run ligand MPNN on the full sequence. You can run Calibre on the full sequence. You can run ESM on the full sequence. So take your pick. And so now you have one backbone, and then modeled on is the sequence and possibly the side chains for that same backbone, different sequences for this population. So we can say we have 10. Um, and then I have two probabilities. So I have two objectives. And so one is the probability of the metal, and the other is probability of the sequence. And we just like you just run those two models with your given sequence and structure. And so I would give my protein sequence, and structure to ligand and PNN and ask it what is the probability of the sequence fitting this backbone, and it would give me some probability. And I would run it with metal 3D, and I would have it try to figure out would there be a metal that exists in this sequence and structure coupling, where would it be, and what's the probability of that metal existing there. So those would be my two probabilities, and I would have those two probabilities for my starting population of 10. And then there's a genetic algorithm approach, and so it has a way of actually selecting out what is the elite. So which ones have the higher probability of both probabilities. So it's like kind of doing this like anomalous like filtering and trying to push up this Pareto farm. So you can imagine it as like starting here, and and then there's like all these other candidates, and it's selecting the ones at the top here. And so then from there, you would get like, you'd have your elite population. And so you take the sequences from them, and then you take like half of the sequence from one, and then just like transplant it into another, and then take half of the sequence from that other, and then translate it back into the one. And so you just cross them over. And then with ligand MPNN, I have it actually just like make mutations. So it selects at random some position or positions, and it will make a mutation there. And then it'll reevaluate this new population in respect to the initial population. What are all the probabilities? And then it would select the new like elite, so like the best scoring ones. And it just continually enriches in this kind of approach, similar to how like nature evolves good function. And so I guess the difference here is that rather than how nature typically does it, we're doing this on a protein structure and protein sequence. And so I have these two probabilities, one for metal and then one for ligand and PNN, but you can have like 5 probabilities, and I could have like a hydrogen bonding probability, and I can have like a score of like how well does AlphaFold like this sequence or something like this. And it's just doing this multi-objective scoring. It's doing the same filtering, enriching for like a population that is happy with all of those criteria. Amelia, does that answer your question a bit more? Perfect. Anyone else? But I don't know if I cut you off.
Cool. I guess we already addressed all the questions. Great.
Okay. Anyone else? I guess we can stop the recording, and then if someone wants to ask a question that is not that they don't want recorded.